Hi all, welcome back to Microbiology Lectures. And now we're going to switch over to talking about eukaryotic microbes. Okay, so we spent a lot of time talking about the prokaryotic microbes, bacteria and archaea. Now we're going to switch and talk about really three kingdoms. We're going to talk about Protista, we're going to talk about Animalia, a few, and then we're going to talk about fungi. And we're just a lot of this is going to be kind of just hitting the surface on a lot of these groups. So I mean, sometimes I'll not talk about many of the microbes that occur in that group, just because either they're not medically important, given the structure of the course. There's a lot of students that are, geared towards medical careers, and so things like the different types of algae and whatnot that may not pertain directly to fields of study, I'm going to skim over them a little bit, so I won't go into great detail. This one, however, we will talk about. This is a picture of Giardia. It's near and dear to my stomach, I guess you could say. I've been infected with Giardia before, and we have kind of a love-hate relationship. Clearly, it's not something that you want to be infected with, but it's a really interesting microbe, and well worth studying and paying attention to. And we'll come back and we'll talk more about some of these different microbes. First thing I want to talk about is kind of the history, or another little piece of history when it comes to microbes and how society reacts to microbes, or in some cases outbreaks, or in this case a famine. Talk about the Irish Potato Famine. It was really a time period of concern from about 1845 to about 1852 or so, but I would say 45, 46 into 47 were the worst for the famine, and this is when the majority of individuals died, and the majority of individuals actually left Ireland. Okay, and so the famine was caused by Phytoplethora infestans, which is a mold. It's a fungus, and so you'll hear me talk about fungi. So mold is not really a type of fungus. It's more like a morphology of fungus, and so it occurs in both mold and spore form. And so the spore form is often able to lie dormant in different regions, and in the case of this species, it lies dormant typically in the soil, and then can be reactivated when conditions are right, and form the hyphae and form the mold and start affecting things like potatoes. And so this infestans tends to ruin crops even today, creates quite a bit of damage to crops, potato crops across the world. But in 45 and 46, it was a really big deal. About 1.5 million people died during the famine. That's not because of consuming infestans and dying from that, although early on, probably in 45, people were eating the potatoes that had infestans on it, so the rotting crop, and they were getting sick and dying from it. But later on, they knew not to eat the infestans or the ruined crop. But the problem is in Ireland, as in many countries in the 1800s and prior to that, once you found a good crop, once you found a good source of nutrients or nutrition, you tend to overgrow that, and you tend to rely heavily on that. And that's exactly what the people of Ireland did during this time. Everyone grew potatoes, and in fact, the poorer portions of Ireland, that was pretty much their entire nutrient source was from potatoes. So when everyone's crops were ruined by this species of fungi, then you start to see huge amounts of starvation. Couple that with some other policies that were going on. Ireland was part of, Ireland was controlled, or at least dictated, by England, and at this time, a lot of their goods were being sent away. So things other than potatoes, so their grains and their livestock were being sent away for finances. And so when the famine hit, those contracts were still abided by. So Ireland still needed to send their resources to other countries, even though they needed their resources to stay in their own country, and they needed, other sources of food, grain and livestock and things like that. But they were obligated and held to the original agreement that they would send all their food away. So basically, they were still able to grow corn and livestock and stuff like that, but they had to send it away due to prior agreements. And so that really created a perfect storm for this species of fungi coming in, ruining all the crops, and then the fact that you didn't have anything else to consume. And so there's lots of sketches and artists and that have depicted this great famine that occurred in Ireland. And I'm not telling you that this didn't happen in other countries. Other countries were affected by it too. Like Phytophthora, it definitely affected pretty much all of Europe that grew potatoes because of the wet, damp weather that was just perfect for this species of fungi. It affected many other countries. It just happened to be that a lot of those other countries, they had other food sources, so they didn't starve. And so when you're completely reliant on a single food source, that's what makes famines extremely hard to get through. And so again, the infection is transmitted by spores that can persist in the soil for a very long time. Still currently today, okay, which we call this potato blight, okay, it's just this mold or fungus that can grow on potatoes, destroys the crop. Still, even today, we lose a lot of money to this crop. Hopefully in the future that won't be the case with new genetic techniques like CRISPR and other possible gene drives. It might be possible for us to alter the genetics of certain species of potato that are really affected by infestants to be resistant to it. And so that is potential in the near future. I know there's a lot of laboratories that are working on this because it's a huge amount of money that can be lost yearly, depending on obviously the environment, because the environment has to be pristine and perfect for infestants to take root or to develop hyphae and become an infective agent. And so sometimes it's dry and the conditions aren't right, and those spores just persist in the soil year after year after year until conditions are great, and then it ruins entire fields of crop. Okay, so we'll look at this from a perspective of how do the fungi do it, like how do they persist, how do they come about. Okay, and so we've talked about this before a while back, that eukaryotic microbes kind of fall into four main groups. Okay, so you have your algae, which are normally protozoans. That's what we're talking about, although some algae are classified as plants sometimes, but algae in general are normally classified as protozoans. Well, and some algae are also classified as lichen because they have a symbiotic relationship with fungi, which we'll talk about later. But these are photosynthetic organisms. Okay, fungi are heterotrophic organisms. Protozoan, unicellular heterotrophs that aren't fungi, so they don't have a chitin cell wall. Okay, algal material. Multicellular organisms, normally if we're talking about the animal kingdom, we're talking about arthropods that are known as vectors. So we're not actually interested in we're not actually typically interested in the fleas, mosquitoes, leeches. That we're interested in what are they capable of transmitting. Okay, are they transmitting a protozoan? Are they transmitting a virus? Are they transmitting a bacteria? So these are transmittive agents, and that's why in micro we'll study like mosquitoes and whatnot. So, for example, in certain species of mosquitoes, they have the ability to carry in their saliva different microbes. Okay, and one that we'll talk about when we talk about protozoans is malaria. And so the sporocytes of malaria typically can reproduce inside the saliva of certain species of mosquitoes, and then when the mosquito bites an individual, it pushes the saliva in because it has an anticoagulant property in their saliva, so the blood can't coagulate and they can take a blood meal. By pushing it in, it pushes in also these protozoans. In this case, Plasmodium would be pushed in to that wound of whatever the organism is. Okay, like lots of organisms can carry different species of malaria. There's lots of different species of malaria. Just some of them happen to be human, transferred to humans, and a concern with humans. And then worms, there are like endoparasites that we often talk about, like tapeworms and flukes and whatnot, pinworms, that we talk about in micro mainly because of the health concerns that are dealing with it. And then often a lot of the cycle of those animals is microscopic. So like they have microscopic cysts that we might need to see in fecal material or whatnot. So they have a microscopic form that's important for identification, or maybe treatment, or at least not allowing the organism to pass from one organism to another. Alright, so obviously we kind of talked about this before, but I'm just going to kind of shoot right over it. Eukaryotes differ from prokaryotes because eukaryotic organisms have a nucleus. They have membrane-bound organelles, or the potential for membrane organelles, given the fact that a nucleus is a membrane-bound organelle. They don't have to have any other membrane-bound organelles, just a nucleus. They don't have peptoglycan in their cell walls, and majority of the time they have a well-developed cytoskeleton. So if you're thinking of cyto as cytoplasm, okay, the jelly-like material inside the cell, the cytoskeleton is the scaffolding, the structure that you would find inside that cytoplasm, which is normally some form of protein, microtubules, other filaments that kind of mesh together to form some kind of apparatus, which can be useful for many ways. It can allow the cell to maintain its shape. It can allow the cell to change its shape, but it also can allow for transport of things like proteins and lipids and other packages that might be self-made along some of these structures to move it throughout the cell. And it's also, it creates a way for things to be stable inside the cell and not kind of float around in the cytoplasm. So you might have some kind of structure like that. Eukaryotes can be considered in many forms, haploid, diploid, polyploid. They have very complex life cycles at times, and many of the species that we'll talk about, they can be haploid, diploid, they could be polyploid, and it really just depends on when you look at the organism and like what time in their life cycle. Like, are they inside the intestinal tract of an organism, or are they in the fecal material? Are they in a body of water, or are they inside the intestinal tract? Are they in the saliva of an organism, or are they in the bloodstream of an organism? So often, this will determine whether or not they're haploid, diploid, polyploid, etc. Also along that is whether or not they're doing asexual reproduction or sexual reproduction. Again, lots of eukaryotes have many different forms of reproduction. They can both do asexual reproduction, typically through mitosis, although some asexual reproduction in eukaryotes is called binary fission. And so it's a little different than mitosis. But sexual reproduction means that they're recombining or using meiosis to make new versions of their chromosomes. Okay? And so sometimes these life cycles are kind of interesting in the sense that you have a haploid organism, okay, so like a single copy of their chromosomes, however many chromosomes they have, and it might be a functioning adult-like organism where they're feeding, maybe they're heterotrophs and they're engulfing things or whatnot. They're carrying out their normal function as an organism, and then conditions are right for them to produce gametes, and those gametes will fuse and go through meiosis, and then you produce a different type of adult, a different type of organism that may carry out very different functions. And so that's some of the cool things that when we look at eukaryotes, they can have these different life cycles that are dependent on conditions. Also makes it somewhat difficult at times to distinguish species, because you might name the haploid version something and then discovered a diploid version of it and not put two and two together that it's the same. And this happens a lot in fungi. Or it used to, prior to the genetics. And again, other things that really connect eukaryotes to prokaryotes are things like we see in protozoans, where they have, two types of nuclei. They have a micronucleus and a macronucleus. So it's similar to a plasmid and the single chromosome of a prokaryote. So prokaryotes have that single chromosome inside a nucleoid region, and then they have these plasmids, which is also genetic material that provides them lots of different advantages, potential advantages in different environments. You have a very similar process or similar structure in some protozoans, where you have these macronuclei, which is the nucleus that's building the majority of the cell or the structure of that organism, and then you have these micronuclei that might just give them this a little bit of an advantage in a different environment. And so through conjugation, they can swap genetic material and recombine, and it's kind of neat systems, but also, again, makes it very difficult sometimes to classify these organisms, because they can swap genetic material more readily than a lot of eukaryotes that might belong to the animal kingdom, and whatnot. Okay. Next time we're going to come back and we'll talk about fungi, and go through the classifications and characteristics of fungi.